So this is on SOS response. And what exactly is SOS response? So SOS response is whenever there is a damage to DNA in the bacterial cell, it is an emergency situation. It is an emergency situation in the organism, and it is attended on top priority. Repair of this damaged DNA is given the topmost priority in the bacterial cell. This response of the bacteria, this response of the organism to DNA damage, it is called as SOS response. Okay. So definition wise, what is SOS response? So you must be able to explain this. This SOS response, it is the response of the organism to DNA damage. Okay, and it is a very swift response. It is attended, DNA damage to the DNA is attended on top priority. So the response of the bacteria, the response of the organism to damage the DNA is called as SOS response. The question that comes next is why? Why it is an emergency and why it is attended on top priority? And the answer is, all of you know by now that DNA is a master molecule. It is a master molecule. Why it is a master molecule? Because it stores genetic information. You all know now that in majority of the organism, DNA is the genetic material. Not only it stores, but it carries developmental blueprints. Developmental blueprints means the genes. Genes are made up of DNA. So any organism's developmental blueprints, that means the entire genome, it is made up of DNA. Okay. And what happens when the DNA gets damaged? The normal replication and transcription processes in the cell stops, because those enzymes which are involved in replication and transcription, DNA pol3 and RNA polymerase, they cannot get past the damaged portion of the DNA. So wherever there is a lesion, wherever there is a damage to the DNA, those enzymes will stop there. They cannot get past, they cannot go through the damaged portion. So what happens when there is a damage to the DNA, the normal replication and transcription processes will stop. And it is for these reasons that damage to the DNA is attended on top priority. That damage to the DNA serves as an emergency in the bacterial cell. Fortunately, on the other hand, what do we have? On the other hand, we have, so all of you know by now that DNA is a sturdy molecule. That means damage to the DNA cannot take place that easily. So we have studied it, we have learned it in one of the lectures that DNA is a pretty sturdy molecule. Okay. Moreover, it is replicated by a high fidelity process. High fidelity process means, you know now that the enzyme which is involved in DNA replication has got built-in proofreading activity. So the chances of error creeping into DNA, newly synthesized DNA, are pretty rare, because the enzyme which is involved has got a rare characteristic of proofreading what it has synthesized. So it is replicated by a high fidelity process, high fidelity process, right? Moreover, structurally, this complementary double helical structure make it easy to retrieve the information as long as one of the strand is intact. If only one of the two strands has been damaged, so because of the complementary double helical structure, it is always easy to retrieve the information. It is always easy to synthesize the damaged strand, because you know what needs to be synthesized. The information is there on the other strand, right? So see the importance. See why nature has evolved this complementary double helical, provided this complementary double helical structure. Right? So complementary double helical structure make it easy to retrieve the information. And the last but certainly not the least, nature has provided excellent DNA damage recognition and repair mechanism to every organism. So every organism has got excellent DNA damage recognition in the first place. And once the damage has been recognized, how to repair that particular damage. So we have already studied, pyrimidine dimer formation and how, cells, how bacteria, they can correct this dimers, pyrimidine dimers. So when the bacterial cell or the DNA gets exposed to UV light, pyrimidines in particular, they have a tendency to form dimer. In pyrimidine also particularly, we have seen thymine dimer formation. So thymine dimers are formed and that serves as a bottleneck. The enzyme cannot get past these dimers. So these dimers are repaired. We have seen base mismatch repair mechanism. So whenever, in spite of all these precautions, all these built-in quality checks, some mismatch of bases can take place. So if that mismatch is there, you got an excellent system. The bacteria has got an excellent system. It can repair that mismatched base. Right now, for ease of operation, of this SOS machinery, SOS mechanism, what nature has done, it has put the genes So for ease of operation, what nature has done, it has put those genes which are involved in the DNA damage repair under common control, so that whenever required, all of them can be expressed, they can be switched on simultaneously, and when not in use, when not required, they can remain shut off. They can remain switched off, right? So what nature has done, probably it has, there are, varying number of figures. Some say that there are 15 genes which are involved in DNA damage repair. Some other book says there are 20. Some others say still higher number. So let us assume that there are approximately 20 genes are involved in DNA damage repair. So what nature has done, nature has, grouped them into 2, 3 operons. So you know what is operon. So there is a promoter and you have these genes here, right? So nature has prepared, nature has grouped them, clubbed them into 2, 3 different operons, or what you call them as replicons. So that as and when required, they can be simultaneously expressed, and when not in use, they can remain switched off. Okay, so when not in use, this promoter is occupied by a protein, by a repressor protein. This is a repressor protein. The name of this repressor protein is LEX-A. So you have to remember this. The name of this repressor protein is LEX-A. This protein occupies the promoter so that RNA polymerase cannot bind over here. And under normal circumstances, when there is no damage to the DNA, these genes will not be transcribed. They will remain switched off. Okay, so the promoter is occupied by a protein, by a repressor protein called LEX-A. Okay, so let us first understand what are the different genes. We are talking about 20 genes. So what are those genes? So you can remember there are genes like, for example, UVRA, UVRB, UVRC. These are the 3 genes which are involved in nucleotide excision repair. Nucleotide excision repair. So we have studied this UVRA, UVRB, UVRC, and these are involved in what is termed as nucleotide excision repair. Similarly, you have genes called UMUC, UMUD. There is another gene called DINB. these genes, they code subunits of DNA Pol 4 and Pol 5. These are the DNA polymerases. These are the DNA polymerase enzymes. So we have seen only DNA Pol 1, Pol 2, Pol 3, but now these enzymes are also known. So these genes, they code for subunits of DNA Pol 4 and DNA Pol 5. Okay, and these DNA polymerases, they do not have proofreading activity. Okay, they can get past those damaged regions. Okay, and because they don't have proofreading activity, any synthesis of DNA done by these DNA Pol 4 and DNA Pol 5, it is prone to higher number of mistakes, higher number of errors, leading to mutations. Okay, so then organism carries out a risk benefit analysis. So there is a risk that when the DNA is synthesized by Pol 4 and Pol 5, there is higher chances of errors leading to mutation. This is the risk, but the benefit is, so if say replication cannot take place, it is going to die. So it is better to go for replication with errors using these enzymes. So you have these genes, they are also DNA damage repair genes. In addition to this, you have SUL-A, SULA, and SUL-B. These genes code for proteins which are cell division inhibitors. So these genes codes for proteins which will not allow the cell to divide because DNA has not been replicated fully. So they will delay the cell division process. So these are all survival tactics, right? So these are some of the common genes which are involved in DNA damage repair. There are approximately 20. And what nature has done, nature has grouped them into maybe two, maybe three different replicons and put them under common control so that as and when required, their synthesis, their expression can take place from a common promoter. Okay, and when not in use, these genes remain switched off because the promoter is occupied by a repressor protein called LEX-A. By a repressor protein called LEX-A. Right? Let us see something about this repressor protein LEX-A. So what is this repressor protein? What are its characteristics? So it's a dimer of 22 kilodalton subunit. It has got approximately 220 amino acids. And there are two distinct domains. The N-terminus domain, N-terminus serves as a DNA binding domain. And the C-terminus, it is involved in dimer formation. Okay, so it reminds you of what? It reminds you of bacteriophage lambda repressor. It is similar to that bacteriophage lambda repressor. This LEX-A repressor, it also has two domains. The N terminus, it is with this terminus, it binds to the DNA. That is called as DNA binding domain. And a C terminus, it is involved in dimer formation, right? And where does it bind? It binds to a 20 base pair consensus sequence in the operator region, and that is called as SOS box. Where does it bind? So in the promoter, we have seen this. So there is a promoter, within the promoter there is an operator, and these are the genes. Right? So somewhere here, you have this SOS box, which is a 20 base pair consensus sequence. It is to this particular region, this 20 base pair sequence, this Lexe repressor protein goes and bind. So when it is bound, it physically obstructs the binding of RNA polymerase, and as a result of that, the genes will remain switched off, right? In addition to Lexe, another protein which plays an important role in this whole process is called as REC-A protein. REC-A protein. So something about REC-A. This REC-A is a 38 kilodalton protein. And it is involved in couple of DNA damage repair activities. And what are those activities? Number one is DNA strand substitution. Substitution, what is referred as recombinational repair. And in SOS response, it serves as a coprotease. Okay, this 38 kilodalton protein, REC-A protein, filamentous protein, it has got couple of DNA damage repair activities. The first one is DNA strand substitution. The damaged DNA strand substitution, it mediates that particular activity, what is referred to as recombinational repair. Recombinational repair. And it is also involved in this SOS response, the one that we are studying right now, by serving as a coprotease. So we'll understand what is meant by this. Okay, so it has multiple DNA binding site. Binding sites, and it binds tightly and in clusters to form what is called as nucleoprotein complex, nucleoprotein filaments. Please understand what I am saying, that this protein has got a very high affinity for the DNA. It has got multiple DNA binding sites and it binds tightly and in clusters, in groups, forming nucleoprotein filament. So as and when the DNA gets damaged, you have a single stranded DNA, it binds to that single stranded DNA tightly in groups, forming what is called as nucleoprotein filament. It has got a built-in DNA dependent ATPase activity. Built-in DNA dependent ATPase activity. In eukaryotes, in higher organisms, its counterpart is called as RAD51 protein. RAD51 protein is the eukaryotic counterpart of REC-A protein in bacteria. REC-A protein बैक्टीरिया का जो यूकेरियोट्स के अंदर काउंटरपार्ट है उसका नाम है RAD51 protein. Okay, this REC-A protein has been the target of many drugs. Because if it is inhibited, if the drug can target it and inhibits it, so DNA damage repair, the damaged DNA cannot be repaired. The DNA damage caused by antibiotics cannot be repaired. So by incorporating that particular compound, the one which inhibits the REC-A protein, what one can do is increase the potency of antibiotics. So antibiotics cause damage to the DNA, but cell has got a built-in DNA damage repair machinery involving REC-A protein. So if REC-A protein is inhibited, the damaged DNA cannot be repaired. So the drug will have a higher potency, right? Now let us see what is the mechanism. So now we know that under normal circumstances, when there is no damage to the DNA, the operons of DNA damage repair genes are switched off. And what causes that switching off? Because the promoter is occupied by, the operator is occupied by Lex A. So under normal circumstances, those genes are not required, their gene products are not required, the proteins are not required. So the operons are switched off. So I am using operons into bracket S. So there may be more than one operon, there may be more than one replicon, there may be more than one group of these genes, right? So as and when there is a damage to the DNA, and you have single-stranded DNA, this SOS response is triggered. This single-stranded DNA combines with REC-A. So we have just studied what is meant by REC-A protein, and they form what are called as nucleoprotein filament. Right? And then this nucleoprotein filament means the ssDNA plus REC-A protein complex. Then activates a latent protease activity in Lex A. So Lex A has got a protease activity which is dormant. But this ssDNA REC-A protein complex, it activates that latent protease activity in Lex A repressor. And it autocatalyzes Lex A, autocatalyzes its digestion. Initially, it was thought that it is the REC-A which has got the protease activity, and it is responsible for cleaving the Lex A repressor. But somewhere in mid-80s, it was found out that it is not the REC-A, rather it is the Lex A itself. It has got a latent protease activity which gets activated in presence of that nucleoprotein filament, in presence of ssDNA REC-A protein complex, and this Lex A autocatalyzes its own cleavage, its own proteolysis. What does it do? It cleaves a particular bond between alanine 84 and glycine 85. Alanine 84 and glycine 85. Alanine at position number 84 and glycine at position number 85, that peptide bond between the two is broken. And as a result of that, the Lex A, it vacates that operator position. And as soon as the operator is vacated, RNA polymerase can bind over there. Okay. So what I'm making it clear is that initially it was thought that it is the REC-A which has got the protease activity, but somewhere in mid-80s, it was found out that REC-A just activates the latent protease activity in Lex A repressor. And this Lex A repressor autocatalyzes its proteolysis, its digestion, its hydrolysis by cleaving a peptide bond between alanine 84 and glycine 85. REC-A serves as a coprotease. REC-A serves as a coprotease, right? So during this process, the number of genes activated is proportional to the DNA damage. So not all the genes are expressed simultaneously, but depending upon the extent of DNA damage, the genes are activated. See the prudency in the bacterial system, that not all those 20, 25, whatever number of genes may be there, all of them are not expressed simultaneously. They assess what is the extent of DNA damage, and based on that assessment, the genes are activated. The first to be activated are UVR A, B, C. First to be activated are UVR A, B, and C. Okay. If the damage is more, then the next one to be activated are UMU, C, UMUD, and DIN B. These are the second set of genes, second group of genes which gets activated. And if the damage that is caused to the DNA cannot be repaired by these also, then in stage 3, SUL A and SUL B, these genes are activated. Okay, because these genes code for proteins which can arrest the cell division, which can arrest the codes for proteins which can arrest the division of cell division, right? So they can delay the division of the cell, right? So genes are activated based on the assessment of DNA damage. Depending upon the assessment, the first set of genes to be activated are UVRABC, and the second set is UMUC, UMUD, and DINB. So now you know that they code for subunits of DNA pol 4 and DNA pol 5, and these DNA polymerases, they are involved in DNA replication without any proofreading activity. So the chances of errors, chances of mistakes taking place during replication are considerably significantly higher. As a result of that, mutation may arise, mutation may take place. And in the third set, SUL A and SUL B, these genes, and therefore I said there may be more than one replicon, there may be more than one operon. So depending upon the extent of damage, a particular operon is switched on. The first one to be switched on is UVR A, B, and C. The first one to be switched on is operon containing genes UVR A, B, and C. The second one is UMUC, UMUD, and DINB. And the third is SUL A and SUL B. So it goes like this. So you have, this kind, now you understand what is meant by an operon. So you have the promoter. Within the promoter, there is an operator, and these are the DNA damage repair genes. Okay, and in the promoter, you have a 20 base pair consensus sequence called SOS box, which is occupied by the Lexi repressor. This is the Lexi repressor. Okay, and as a result of that, RNA polymerase cannot bind and transcription cannot take place. Okay, but when the DNA damage gets damaged, so in presence of ssDNA, Rec A complex, which activates a latent protease activity in Lexase, it autocatalyzes itself and the promoter gets vacant. It autocatalyzes, so Lexase gets removed. So the promoter is now vacant and it will be occupied by RNA pol. This is RNA pol. And it will start transcribing the genes. Transcription will take place, followed by translation. Okay, so depending upon the extent of damage, the operons are switched on. The first one to be switched on is the operon which contains UVR ABC genes. The second one is UMUC, UMUD, and DINB genes, which code for subunits of DNA polymerase 4 and DNA polymerase 5. And then if the damage is more, which cannot be brought under control by these two steps, then the third one, it switches on the operon which codes for genes, which expresses the genes which code for inhibitors of cell division. So cell division is delayed. So this whole thing is called as SOS response. So that's all in this. Thank you very much.